actually it's surprising. I really actually was convinced it had to be an overfit. The next idea was internal consistency. Random forest, at the end of the day, is a Monte Carlo process. So the question was, well, how stable are these results? So again, I reran the same process, just changing the random number seed. For computational reasons, this is more for illustrative for myself, since it's only done once. This isn't a full permutation, this isn't a full stability test. But of the top 20 SNPs, 19 of them were also found among the top, again suggesting that there was pretty high stability of the results. And the final test for confidence was just some external consistency. And particularly, I want to compare to that 2007 study, which used the exact same data set to see, well, are we able to duplicate any of their findings? And in an ideal world, you'd want to duplicate some of their findings because it is the same data set. You don't want to do something radically different. At the same time, you don't want to find exactly what they find because, well, then I just spend a lot of hours of work for nothing, which is something I can find by just doing a marginal test. And that's exactly the ideal world that was found. Among the 13 genes, 9 were found among the 3 configurations. They weren't always the top ones. And I was also able to identify some other genes that also showed some promise and interest, which are currently being followed up on in our group. So at this point, just a quick summary and some thoughts. Particularly for genetic data, it does appear that LD pruning is an effective means of reducing data size and can also speed up computation. The HLA appears to dominate all of the potential results, and I think it's something that, a lesson that we can all learn with working with this type of data, that one particular strong association you may gain something by taking it out of the analysis. And it does appear that random forest can duplicate some previous findings while also suggesting new ones. And in terms of general approach, it does appear that the default value of mTri is not appropriate, though it is still fairly robust. Subsampling is a fairly good process to go through. And it does appear that different configurations will show slightly different rankings of variables, really suggesting that when doing really any machine learning algorithm, it's a good idea to really play around with the data and see, what can you get from it, because different configurations will yield different results. Remember, at the end of the day, we're not doing hypothesis tests, so we're not really worried about what's the multiplicity issue. We're just trying to find interesting findings. So at this point, if you're a biostatistics graduate student and you do an analysis and you get some interesting results, you're directed to do a simulation study to follow up and try and really understand the results. So that is what I did and what is currently ongoing. And so I'm going to present some data from a simulation study that is currently running on our cluster in our lab. But I'm going to try and address four key questions from this. The first is kind of the meta question of how effective is random forest compared to just doing a marginal test. We find something interesting, but are we finding anything better? Is this work really worth it? The next is, how do these variable importance measures compare? As I recall, all I looked at was just the permutation importance. So what if we looked at the Gini importance? How does the mTri impact the solution? And are there certain types of effects that are easier to find than others? So to do this, I simulated 25 data sets. The reason, again, only 25 was just for some of the computational amount involved. And within each of those data sets, there were 77 causal variables containing different genetic models and correlations. This is simulated via a logistic regression. It is overall, I believe, a 44-parameter logistic regression. And I attached to those causal variables the 160,000 noise variables that came from the LD 90% data set. The reason I used empirical data for the noise was it's really impossible for them to simulate realistic genetic data. There are some packages out there, but it's, at the end of the day, you're never going to replicate the structures that you'll find in genetic data. So each of these 77 causal variables had a different effect type, MAF, and model. So I looked at main effects, two-way and three-way interactions, and these heterogeneous effects, basically effects that affected either a high percentage of the population or a low percentage of the population, different levels of MAF, minor low frequency, and then the 3 different causal model types. The random forest method I grew 2,000 trees with 3 different mTri values. For this, I actually used my own version of random forest that I wrote in C. The reason for this was I was able to optimize it a bit for SNP data, and also from a research perspective, it really allowed me to ask questions and get added information that I needed. And I ranked the variables based on the 2 random forest variable importances as well as their marginal p-value. and then did almost what is essentially a power calculation, though not really. I just compared the number of true hits among the top 50, 100, 1,000, and 5,000. So for example, what I did was if I say I would rank the variables by random forest method and say, among the top 100 hits of random forest, how many of them are true associations? Among the top 1,000, how many of them are true associations? So the first thing that we have here is just looking at the number of true effects by each of the three different importance measures. In these three graphs, we've stratified by the three M-tribe values. And going along the x-axis are the number of top SNPs looked at. The y-axis are the number of true associations. And then in the clusters, we have in the black, we have the p-value, the permutation importance, and the Gini importance. And what should really jump out here are two things. For one, it appears actually that the Gini importance is really dominating the permutation-based importance. And for those who are more intimately familiar with these measures, I can speak a little bit more about why I think that's the case. But actually, this is actually a much better measure to use. But even more so, but what also becomes interesting is that when we're looking at, say, the top 50 or top 100, it appears that the Gini importance is doing just as well, if not better, than ranking by p-value. However, once we start looking at the top 1,000 and 5,000, it starts appearing that the p-values now are moving a little bit further away and doing a little bit better. Another way to illustrate this is that here we have now along the x-axis are the number of top markers looked at, the y-axis are the number of variables picked up, and here it becomes a little bit more clear. In the colored lines are the different M-tri values, and the black line is the p-values. And we find, again, among the top hits, if you just look at the top 50 or 100, p-values are doing just as well, if not worse, than random forest. But once you move to the top 1,000, 5,000, and this becomes even more obvious in looking at the top, say, 10,000 or 50,000, p-values are a much more powerful approach. So this was actually initially a fairly discouraging finding, though it does make somewhat sense. But essentially what's saying is that we're not really gaining, in some ways we're not really gaining all that much by doing all this work. We'd find a lot more true hits had we just looked at marginal p-values. And if we think back to the empirical study and we think about, okay, what were the types of associations that were found, they were at the end of the day real effects. They had marginal associations with p-values around 10 to the minus 3, 10 to the minus 5. So it's not as if we're going to pick up something with this type of algorithm, or really any algorithm, that has a marginal association of 0.5. We're not going to get something for free. Essentially, in some ways, we can think of what this is doing is, what these learning algorithms are doing is re-ranking among what are probably the top associations. Yeah.
I got confused.
Yes.
77 causals in the simulation.
Yes.
What was the structure that you imposed on their activity or stasis?
So it was a logistic regression. So there were some. Yeah, so it was logistic. So there were some that were explicit. So of the 77, it was 44 parameters. So there were some that were actual interactions. An interaction between, variable 1 and variable 2 would just be a single parameter. But at the end, it was just an independent logistic regression. So for each simulated person, you'd get a probability of being diseased, and that became their probability of being a case.
How did you decide how much complexity was built into the interactions of the 77?
How much complexity? I mean, it was just. Each variable in the interaction just had a minor allele frequency and simply whether it was a dominant effect, an additive effect, or a recessive effect. And then it was just a straight interaction of that x1 times x2. So you have an observed x. So you end up with an observed x1 and x2 for each person, and it's just a straight multiplication.
So you built in some two-way and some
three-way, correct? Yeah. Mm-hmm. Well, it wasn't arbitrary. It was pre-decided. So for each of these 25 data sets, the logistic model is identical. So as mentioned, this is kind of a slightly discouraging thought. So the next question that became, and this is, I guess, what I'd say is the take-home point, and where things became kind of interesting, is, well, are we finding different types of effects with these methods? And so there's a lot going on in this graph, and I'm going to try and break it down. What we have is, this is now looking at the Gini variable importance with an M-tri of 10%. We have the three different factors that went into the causal effects. Along the, in each of the groupings, we have the number of top hits looked at, 50 to 5,000. The y-axis is the percentage found. And to give a little bit of context, the black line is what the percentage found was had we just ranked by variable importance.